Hello, and welcome to my presentation of Anaerobic reductive bioleaching of manganese ores. I am Neha Sharma, a master's and PhD student in the Department of Chemical Engineering, Michigan Technological University. This project is funded by the US Department of Energy and advised by Dr. Tim Eisler. This presentation will discuss the significance of bioleaching in case of manganese ores, mechanism involved in the bioleaching process, materials and methods used in this study, results and discussions, and will end with conclusion and future work. So why is bioleaching important in the case of manganese ores? Considerable quantities of low-grade manganese ores are present in the US that are not being mined. Conventional methods are uneconomical and produce pollutants. Manganese is vastly used in the industry and domestically is not produced in the US. Bioleaching can be applied to low-grade manganese ores in an economical and environment-friendly way. Manganese is used in vast quantities in the steelmaking industry and battery making industry. The other uses of manganese include paint and dye industry, glass industry, agriculture industry, chemical industry. In the direct reduction process, mainly bacteria are involved. The image shows the transfer of electron across the surface of a bacterial cell and MnO2 in which manganese acts as a terminal electron acceptor and is thus reduced to manganese in the 2 plus form in the solution. In the indirect reduction of manganese dioxide, microorganisms, which include bacteria and fungi, produce organic acids such as citric acid, oxalic acid, acetic acid, among other acids, which attach themselves to the surface of manganese ores and form a manganese 3 plus complex, which is then reduced to manganese in the 2 plus form with the presence of other acids in the media. In this study, 6 anaerobic flasks containing reagent-grade manganese and high-grade manganese ores were used. 60 ml nutrient media, which was prepared by adding 500 ml cattails decant and 2.5 grams granulated sugar, was applied to the flasks every week and samples were collected. The manganese concentration was estimated by using the periodic method with the help of Hatch test. A manganese leach bucket was also set up in the imitation of the fen from where bacteria were obtained. The results obtained in this study are shown in the graph. Dissolved manganese concentrations of 866.7 mg per liter for reagent-grade manganese dioxide and 385.2 mg per liter for O-grade manganese was found. Manganese leach bucket precipitate contained 12.81% manganese. The pH of the nutrient media was 6.9 and that of the leaching flask was around 5, which indicate the production of acids by the bacterial community. The concentration of dissolved manganese are significant to continue research on varying parameters that affect manganese dissolution. Future work will include the variation of parameters like pH and carbon source to determine optimal conditions for manganese dissolution. Future work will also include the estimation of dissolution of iron after manganese has been preferentially leached out. The image shows manganese leaching setup which is hypothetical and could be used on a large scale.